All right, welcome back. Today we're going to do a deep dive into something that's absolutely fundamental to life as we know it, the cell. We've got this awesome visual textbook on cell biology, full of diagrams and even some online animations. And plus, we've got some expert insights to help us really connect the dots and see how it all works.
Yeah, what's really fascinating about this deep dive is that we're going to see how these tiny building blocks create all the amazing complexity and diversity of living organisms, from the smallest bacteria to those giant redwood trees. That's all thanks to cells.
Yeah, it's kind of mind-boggling when you think about it. Trillions of these little guys working together to create everything we see around us. But let's not get ahead of ourselves. Let's start with the very basics, right?
Absolutely. Got to understand the foundation before we can appreciate the whole structure. So let's begin with those tiny building blocks of matter, atoms.
Okay, atoms. I vaguely remember those from high school chemistry. They're like super small, right?
Tiny, but mighty. Atoms come together to form molecules, and the way they bond determines a lot about how those molecules behave. Strong, covalent bonds, they act like a superglue, holding things together tightly, provide stability. Then you have these weaker interactions, like hydrogen bonds. They're more like temporary connections, but incredibly important for these dynamic processes that are always going on within cells.
So it's like the difference between welding something together permanently versus using magnets that can connect and disconnect as needed. Both have
That's
their uses, right?
exactly. And when we zoom in on those organic molecules, the ones that make up living things, we find four major families: sugars, fatty acids, amino acids, and nucleotides. We always hear about carbs, fats, and proteins when it comes to nutrition. Well, those are built from these families. Sugars are the backbone of carbohydrates, fatty acids create fats, and amino acids, they link up to form proteins. So basically, these four families are like the ingredients list for life itself.
Okay, that's a great analogy. It's making a lot more sense now. But how do we get from these small, simple molecules to the big, complex structures we see in cells?
That's where macromolecules come into play. These are larger molecules that are assembled from those smaller building blocks. Think of it like this. If the small organic molecules are individual Lego bricks, then macromolecules are those amazing structures you can create by putting those bricks together.
So we're talking about things like DNA, proteins, that kind of thing.
Exactly. So, for example, sugars. They can link together to form polysaccharides. They can provide structural support, like in plant cell walls. Or they can store energy, like glycogen in our muscles. Fatty acids, they come together to form lipids, those essential components of cell membranes, which are kind of like the walls and barriers within a cell.
And of course, there are proteins built from those chains of amino acids. I've always been fascinated by proteins. It seems like they do just about everything in a cell.
They really do. They're incredibly diverse and adaptable. Proteins can act as enzymes, which speed up chemical reactions that would otherwise take forever. They can serve as transporters, moving molecules across cell membranes, like tiny cargo ships. And they even act as signal integrators, relaying information within and between cells. It's like they're the ultimate multitaskers of the cellular world.
Wow, so proteins are like the movers and shakers and communicators of the cell, but how do they manage to do so many different jobs? It seems like that would require some serious specialization.
The secret lies in their unique 3D shapes. Imagine this. A protein's amino acid sequence, which is like a long chain of beads, determines how it folds into this complex three-dimensional structure. And this intricate shape is what allows it to carry out its specific function. It's like each protein has a custom-designed toolkit, perfectly shaped to do its job.
So it's not just about the ingredients, the amino acids, but how they're arranged in space. It's like, oh, origami, right? The way you fold the paper determines what you create.
That's a great way to put it. And what's really mind-blowing is that if you unfold a protein, it can often spontaneously refold back into its correct shape. It's like it has a built-in blueprint for how to assemble itself. This demonstrates that the information for a protein's 3D structure is actually encoded in its amino acid sequence. It's all there in the blueprint.
That's incredible. So the sequence of those amino acids is like a set of instructions, guiding the protein to fold into its functional form. It's like having a self-assembling IKEA bookshelf.
Exactly. Now, to understand these intricate 3D structures, we need to talk about the different levels of organization within a protein.
Okay, let's break that down. What are these different levels?
We'll think of it like building a house. The primary structure is the foundation, the linear sequence of amino acids. Then comes the secondary structure, introducing some basic architectural elements like alpha helices and beta sheets. These are recurring patterns that form within the protein chain, adding stability and shaping the overall structure.
So if the primary structure is like the list of materials, the secondary structure is like the framing and the walls of the house.
Exactly. Then we have the tertiary structure, which is the overall 3D shape of the entire protein, like the complete house with all its details and finishing touches. This level of structure is crucial for a protein's function as it determines how it interacts with other molecules.
Hmm. Okay, so it's not just about the individual parts, it's about how those parts come together to create a functional whole. So what about quaternary structure? Is that like an entire neighborhood of
You
houses?
got it.
Yeah.
Quaternary structure describes how multiple protein subunits, each with its own tertiary structure, can interact and assemble into larger, even more complex structures.
So proteins aren't just solo acts. They can team up to form even more powerful molecular machines. That's fascinating. Can you give us some examples of how this structural hierarchy relates to a protein's function in a real cell?
Sure, so let's picture a protein floating around in that watery environment inside a cell. Its nonpolar side chains, the ones that don't like water, cluster together in the core of the protein, kind of like penguins huddling together for warmth. Meanwhile, the polar side chains, the ones that do like water, they're on the outside, happily interacting with the surrounding water molecules.
Okay, I can visualize that. So it's like the protein is organizing itself to stay comfortable in its watery surroundings.
Precisely. Now imagine a protein embedded within a cell membrane, which is like a wall separating different compartments within a cell. The part of the protein that spans the membrane is often an alpha helix, because that shape is particularly good at shielding the protein's backbone, which doesn't like the oily environment of the membrane. It's like the protein is wearing a raincoat to stay dry.
I love that analogy. It makes it so much easier to understand. So alpha helices are like tiny protein raincoats, protecting the protein as it crosses the membrane.
Exactly. And proteins can get even more ambitious with their structural organization.
Yeah.
They can assemble into these long filaments, like actin filaments, which are crucial for cell shape, movement, and muscle contraction. It's like a cellular skeleton, providing support and allowing for movement.
So actin filaments are like the beams and girders of a cell.
Exactly. And then we have microtubules, which are these hollow tubes that act like cellular highways for transport. They help move organelles and vesicles around the cell, kind of like a cellular subway system.
So if actin filaments are the beams, microtubules are the train tracks.
A perfect analogy. And proteins can also form these protective cages, like the coating coats of viruses, which shield the viral genetic material as it travels from cell to cell.
So proteins are involved in just about every aspect of a cell's structure and function. It's like they're the ultimate construction crew, building, maintaining, and even defending the cell.
Absolutely. And their talents don't stop there. Let's dive into the world of enzymes, those remarkable proteins that act as catalysts of life.
Okay, enzymes. I've always been curious about how they work. How do they actually speed up reactions? Without being consumed themselves, it seems like magic.
It's not magic, but it's pretty darn close.
Yeah.
Imagine you have a boulder and you need to push it over a hill. That hill represents the activation energy, the energy that's needed to get a reaction going. Enzymes are like these magical bulldozers that lower that hill, making it easier for that boulder to roll over.
So they make it easier for reactions to happen, but they don't get used up in the process. It's like they're facilitating the reaction without actually participating in it.
Exactly. It's like they're the matchmakers of the cellular world,
Right.
bringing molecules together in just the right way to make those reactions happen more easily.
But do enzymes work on just any reaction? Or are they more specialized?
Oh, enzymes are incredibly specific. They have this particular shape that perfectly fits their target molecule called the substrate. Think of it like a lock and key. Only the right key will open the lock.
So one enzyme, one specific job, it's all about shape and fit.
Exactly. Take hexokinase, for example. This enzyme adds a phosphate group to D-glucose, a specific type of sugar. But it won't even touch L-glucose, which is its mirror image. It's like trying to fit a left shoe onto a right foot. It just won't work.
So it's all about that precise fit between the enzyme and its substrate. That's incredible. But how do cells control all this enzymatic activity? It seems like it would be chaos if enzymes were just running around doing whatever they wanted.
You're right. There needs to be some regulation. And that's where feedback inhibition comes in. It's like a thermostat, keeping things in balance.
Okay, I'm intrigued. How does that work?
Imagine a metabolic pathway, a series of reactions that produces a specific molecule, like an assembly line in a factory. When that final product builds up, it can actually bind to an enzyme earlier in the pathway, essentially shutting down the production line. It's like the final product is saying, hey, we've got enough of me, time to stop production.
So it's like a self-regulating system. That's pretty clever. It's like a built-in quality control mechanism, making sure the cell doesn't waste energy producing more of something than it needs.
Precisely. And this kind of regulation is happening all the time in cells, keeping everything in balance and ensuring that resources are used efficiently.
This is fascinating stuff. But I've also heard about this thing called the Michaelis-Menten equation. What's that all about? Is it some kind of secret code for enzymes?
The Michaelis-Menten equation, well, it's not a secret code, but it is a powerful tool that helps scientists understand how enzyme activity changes with different substrate concentrations. It's like a blueprint for characterizing how efficient an enzyme is at its job.
So it's a way to measure enzyme performance. It's like the MPG rating for a car.
That's a great analogy. It helps us understand how well an enzyme converts its substrate into product, giving us insights into the enzyme's efficiency and how it might be regulated. Now, since we're talking about blueprints, we can't forget DNA replication.
Ah, yes, the ultimate copy machine, the molecule that holds the instructions for life itself. But how does DNA actually make copies of itself? It seems like such a complex process.
It is a remarkable process called semi-conservative replication. Picture the DNA double helix as a twisted ladder. To copy it, the ladder unzips, and each half serves as a template for building a new complementary strand.
So it's like each half of the ladder gets a brand new partner, and the end result is two identical DNA molecules.
Exactly. Each new DNA molecule contains one original strand from the parent molecule and one newly synthesized strand. It's like passing down a family heirloom, but making a perfect copy so that both the parent and the offspring have one.
Okay, that makes sense. But how do scientists actually know that's how it works? Was there some kind of experiment that proved it?
Yes, there was this brilliant experiment called the Michelson-Stahl experiment. Researchers grew bacteria in a special medium containing heavy nitrogen, which got incorporated into their DNA, making it denser than normal DNA. Then, they switched the bacteria to a medium with normal nitrogen and let them divide. By analyzing the density of the DNA in each generation of bacteria, they were able to track where the original heavy DNA strands went.
So it's like they were tagging the original DNA with a special label so they could follow it.
Exactly. And what they found was that the first generation of daughter DNA molecules had an intermediate density, indicating that they contained one old heavy strand and one new light strand. This confirmed the semi-conservative model of replication, proving that each new DNA molecule inherits one strand from the parent molecule and synthesizes a new complementary strand.
Wow, that's a really clever experiment. So they basically caught DNA replication in action, proving that it's a semi-conservative process. That's pretty amazing.
Yeah. It was a groundbreaking experiment that revolutionized our understanding of how DNA replicates, and it paved the way for many more discoveries in molecular biology.
Okay, so we've got the DNA replicated, but what happens next? How does that information encoded in DNA actually get used to build a living organism?
That's where we move from DNA to protein, a process known as the central dogma of molecular biology. It's like taking the blueprints from the architect's office and using them to actually build the house.
All right, let's break down this dogma. What's the first step?
The first step is transcription, which is kind of like copying a recipe from a cookbook. DNA serves as the master cookbook containing all the genetic information, and RNA polymerase is the meticulous chef, carefully copying the recipe. It reads the DNA sequence and creates a complementary RNA molecule called messenger RNA, or mRNA.
So mRNA is like the recipe card, carrying the instructions from the DNA cookbook out to the kitchen, where the proteins are made.
Exactly. But there are other types of RNA too, each with its own specialized role in the cell. Ribosomal RNA, or RNA, is like the kitchen counter, forming the core of the ribosome, the protein-making factory. Transfer RNA, or tRNA, acts like a delivery truck, bringing the correct amino acids to the ribosome, based on the instructions on that mRNA recipe card.
Wow, so it's like a perfectly coordinated system with each type of RNA playing a specific role in translating those DNA blueprints into actual proteins. It's like a cellular ballet.
Yeah, and in eukaryotes, organisms like plants and animals, the process is even more intricate. Before that mRNA can leave the nucleus, the cell's control center, where the DNA is stored, it undergoes RNA processing, gets capped, spliced, and polyadenylated. Think of it like quality control and packaging for shipping.
So the mRNA gets a makeover before it goes out into the cytoplasm to do its job.
Exactly. And this processing is really important for ensuring that the mRNA is stable, can be transported efficiently, and is properly translated into protein. One interesting aspect of RNA processing is alternative splicing.
Alternative splicing, what's that?
It's like having a single recipe that you can modify to create different variations of a dish. Alternative splicing allows a single gene to produce multiple different proteins. By mixing and matching different parts of the mRNA, it's like having a single set of clothing patterns that you can use to create multiple different outfits by combining different pieces.
That's incredible. It means that a single gene can be much more versatile than we originally thought. It's like maximizing the potential of the genetic cookbook.
Absolutely. Alternative splicing allows for greater complexity and diversity in the proteins that can be produced from a limited number of genes, increasing the coding potential of the genome.
So it's a way for cells to get more bang for their buck, genetically speaking. I love how nature is always finding these clever ways to do more with less.
It's truly remarkable.
Right.
Now, once that processed mRNA reaches the cytoplasm, the real show begins, translation, the process of actually building a protein.
Okay, time to cook up some proteins. How does this translation process work?
Think of the ribosome as the kitchen counter, the mRNA as the recipe card, and the tRNAs as the ingredients. The ribosome reads the mRNA, codon by codon, which are like three-letter words that specify each amino acid. Each tRNA, carrying a specific amino acid, binds to the mRNA based on its codon. And the ribosome links the amino acids together, like assembling a complex Lego structure one brick at a time to form a polypeptide chain.
So the ribosome is like the master chef, carefully following the recipe on the mRNA card, and the tRNAs are like sous chefs, bringing the right ingredients at the right time.
That's a great analogy. And this whole process, from transcription to translation, relies on the genetic code, which is like the universal language of life. It's a dictionary that relates each codon to a specific amino acid, ensuring that the protein is built correctly.
It's incredible how nature has come up with such an elegant and efficient system for building proteins, but I imagine there must be some way to control which proteins are made and when, right? It can't just be a free-for-all in the cell.
You're absolutely right. Cells have to be very selective about which genes are expressed, or turned on, and when. It's like having a vast library of recipes, but only choosing to cook certain dishes at certain times, depending on the occasion and the available ingredients.
So there must be some kind of control mechanism, like a cellular librarian. Who decides which books or genes are available to be read and translated?
Exactly. And that's where gene regulation comes in. It's like a conductor leading an orchestra, ensuring that the right instruments are playing at the right time and at the right volume. Gene regulation ensures that only the necessary proteins are made, in the right amounts, at the right time.
That makes sense. It would be a waste of resources if every gene was being expressed all the time. So how do cells actually control which genes are on and which are off?
Well, one of the key players is transcription regulators, proteins that bind to specific DNA sequences near the start of a gene. Think of them like dimmer switches for genes. They can either boost the expression of a gene, turning it up, or inhibit its expression, turning it down, or even turning it completely off.
So these transcription regulators are like the volume knobs for genes, controlling how much of a particular protein is made.
Precisely. And there's another level of control: chromatin structure. Chromatin is the complex of DNA and proteins that make up chromosomes. The way DNA is packaged within chromatin can influence gene expression. It's like making the cookbook more or less accessible to the chef. If the DNA is tightly packed, the chef can't read the recipes. But if it's loosely packed, the recipes are easy to access and use.
So it's like having a really organized bookshelf where you can easily find the cookbook you need, versus a chaotic pile of books where it's hard to find anything.
A perfect analogy. And this packaging of DNA is influenced by modifications to the histone proteins that help organize the DNA. It's like adding bookmarks or sticky notes to the cookbook, marking certain recipes as important or highlighting sections that shouldn't be used.
That's fascinating. So there's this whole other layer of control happening at the level of DNA packaging. It's like the cell is carefully curating its genetic library, making sure that the right information is available at the right time.
Exactly. And this regulation of gene expression is incredibly important for cellular function, development, and even disease. It's what allows cells to specialize and carry out their specific roles in the body.
This is all so incredibly intricate and complex. It's amazing to think about all the different layers of control and regulation that are happening within a single cell. And it makes you realize that even though cells are tiny, they're incredibly sophisticated and dynamic systems.
You're absolutely right. And we've only just scratched the surface of this amazing world of cell biology. There's so much more to explore, so many fascinating mechanisms and processes yet to be discovered.
Well, I'm definitely hooked. I can't wait to dive deeper and learn more about how cells work their magic. Where do we go from here?
From here, we can delve into how cells maintain their genetic integrity and pass on that information to future generations. This involves a tightly regulated process called the cell cycle.
The cell cycle. Isn't that how cells replicate themselves?
Exactly. Think of it as a carefully choreographed dance routine, a series of steps that a cell goes through to duplicate its DNA and divide into two identical daughter cells.
So it's not just about copying the DNA, it's about making sure everything is in order for a smooth and successful cell division.
Precisely. The cell cycle is divided into these distinct phases, G1, S, G2, and M, each with its own set of tasks and checkpoints to ensure everything proceeds smoothly.
Okay, walk us through those phases. What happens in each one?
G1 is like the warm-up, a period of growth and preparation for DNA synthesis. The cell is gathering resources, making sure everything's in place for the big event DNA replication.
So it's like setting the stage, making sure the actors have their costumes and the lighting is just right before the curtain rises.
A great analogy. Then comes S phase, where the main act takes place. This is when DNA replication happens, with each chromosome creating a copy of itself. It's like doubling the amount of genetic material, so that each daughter cell will receive a complete set of instructions.
Okay, so S phase is when the magic happens, the DNA is copied, the genetic information is duplicated. What happens after that?
After S phase, we move into G2, which is kind of like a dress rehearsal. It's a quality control phase where the cell checks for any errors in DNA replication and makes any necessary repairs. Think of it like proofreading, a really important document. Before hitting send, you want to make sure there are no typos or mistakes that could lead to problems later on.
That makes sense. It's like a final check to make sure everything is in order before proceeding with the grand finale of cell division.
Exactly. And then finally, we reach M phase, the grand finale itself, which includes two main events, mitosis and cytokinesis.
Mitosis, the division of the nucleus, and cytokinesis, the division of the cytoplasm. It's like the climax of the dance where everything comes together.
During mitosis, those duplicated chromosomes are carefully separated and distributed to the two daughter cells. It's like a perfectly synchronized ballet, with each chromosome moving to its designated spot with incredible precision.
I can almost picture those chromosomes gracefully gliding across the cell, taking their positions for the final split. It's amazing how cells can manage such complex movements with such accuracy.
It's truly remarkable. Mitosis is orchestrated by these spindle fibers, which are made of those microtubules we talked about earlier. They attach to the chromosomes and guide them to opposite ends of the cell, ensuring that each daughter cell receives a complete set of chromosomes.
So it's like a carefully planned tug of war, making sure each daughter cell gets an equal share of the genetic inheritance.
Right, and then cytokinesis completes the process, dividing the cytoplasm and pinching the cell in two, creating two distinct daughter cells, each with its own nucleus and a full complement of chromosomes.
So from one parent cell, we get two identical daughter cells, each ready to embark on its own cell cycle journey. It's like a perpetual dance of life, with cells dividing and creating new cells in a never-ending cycle.
And this entire process, from that initial growth phase to the final division, is tightly regulated by a complex network of proteins. Key players in this regulatory network include cyclins and cyclin-dependent kinases, or CDKs.
Okay, cyclins and CDKs, those sound familiar. Can you remind us what they do?
Think of CDKs as the engines that drive the cell cycle forward, and cyclins as the gears that control their speed and direction.
So it's like having an accelerator and steering wheel for the cell cycle, controlling when it starts, how fast it goes, and when it stops.
Exactly. Cyclin levels fluctuate throughout the cell cycle, binding to and activating specific CDKs, which then act like switches, turning on or off the various events of each phase. It's like this carefully timed sequence of events, with each cyclin-CDK partnership controlling a specific part of the dance.
So it's not just about blindly going through the motions, it's about having this sophisticated control system to make sure everything happens at the right time and in the right order.
Precisely. And to add another layer of control, there are checkpoints throughout the cell cycle, like quality control inspectors, making sure everything is proceeding correctly before moving on to the next stage.
So it's like having multiple inspection points along the assembly line, making sure each part's properly built before moving on to the next step. It's a very thorough process.
These checkpoints are crucial for preventing errors and ensuring the integrity of the genetic information being passed on to the daughter cells. They can pause the cell cycle if there are problems, like DNA damage, giving the cell time to make repairs. And if the damage is too severe to repair, these checkpoints can even trigger a process called apoptosis.
Apoptosis. Programmed cell death. It sounds a bit dramatic. Why would a cell choose to self-destruct?
It might seem counterintuitive, but apoptosis is actually a vital process for maintaining the health and integrity of an organism. It's like a built-in safety mechanism that eliminates cells that are damaged, malfunctioning, or no longer needed. It's like demolishing a building that's become unsafe or outdated to make way for new construction.
So it's not always about dividing and multiplying. Sometimes cells need to sacrifice themselves for the greater good of the organism.
Exactly. Apoptosis plays a crucial role in development, tissue homeostasis, and preventing diseases like cancer. It ensures that only healthy and functional cells contribute to the organism's survival.
That makes a lot of sense. It's like a cellular quality control system, making sure that only the best cells survive and contribute to the organism's well-being.
Now, speaking of cell death and renewal, let's shift our focus to tissues and stem cells.
Okay, so we're moving from single cells to multicellular organisms. How do individual cells come together to form tissues and organs?
Well, in multicellular organisms, cells don't just exist as individuals, they work together in these organized communities called tissues, just like a well-coordinated team. Each cell in a tissue has a specific role to play. Think of muscle tissue, for example. It's made up of cells that can contract, allowing for movement. Then there's nervous tissue, which is specialized for transmitting signals throughout the body. And epithelial tissue forms protective barriers like our skin.
So it's like different departments in a company, each with its own specialized function, working together to achieve a common goal. And then these tissues come together to form even larger structures, like organs, right?
Exactly. Tissues work together to form organs, which are like complex machines with multiple parts working in harmony. Your heart, for example, is an organ made up of muscle tissue, nervous tissue, and connective tissue, all working together to pump blood throughout your body. Your lungs are organs that take in oxygen and release carbon dioxide, thanks to the specialized epithelial tissue that lines their tiny air sacs. And your brain, the control center of your body, is this complex organ made up primarily of nervous tissue.
It's amazing to think that all these different organs, each with its own unique structure and function, arise from a single fertilized egg. But how does this incredible organization and specialization happen? How do cells know what to become?
That's where cell differentiation comes in. It's the process by which cells become specialized, taking on specific roles and characteristics. Think of it like students choosing their majors in college, each pursuing a different path based on their interests and abilities.
So even though all the cells in your body have the same DNA, they can become incredibly diverse through this process of differentiation.
Exactly. Cell differentiation is driven by changes in gene expression, those patterns of gene activity that we talked about earlier. Different cells are essentially reading different parts of the same genetic cookbook, expressing different sets of genes, which leads to the production of different proteins and the development of different characteristics.
So it's like having a master recipe book with instructions for making all the different parts of the human body. But each cell only uses the recipes that are relevant to its specific role.
A perfect analogy. This selective gene expression allows cells to acquire specialized structures and functions, creating the incredible diversity of cell types we see in multicellular organisms. It's what allows a single fertilized egg to develop into a complex organism with trillions of cells, each playing its part in the symphony of life.
This is all so mind-blowing. But what about cell renewal and repair? Our bodies are constantly replacing old and damaged cells. How does that happen?
That's where stem cells come in. They're like the ultimate multitaskers of the cellular world. Stem cells are undifferentiated cells that have this remarkable ability to self-renew, meaning they can divide and create more stem cells, and to differentiate, meaning they can develop into specialized cell types. They're like the blank slates of the cellular world, with the potential to become any type of cell in the body.
So they're like the cellular equivalent of those multi-talented people who can do just about anything. Write, sing, dance, build, you name it.
That's a great way to put it. Stem cells are found in various tissues throughout the body, like bone marrow, skin, and even the brain. They act like a reserve of fresh recruits, ready to be trained for specific roles as needed.
So if a particular tissue needs more cells, stem cells can step up and differentiate into those specific cell types.
Exactly. Stem cells play a vital role in growth, repair, and maintenance throughout our lives. They replace cells that are lost due to injury, wear and tear, or just the normal aging process. They're essential for keeping our bodies functioning properly.
It's incredible how our bodies are constantly renewing and repairing themselves. But I imagine there must be different types of stem cells, right? Not all stem cells have the same potential to differentiate.
You're absolutely right. Stem cells vary in their potency, which is their ability to differentiate into different cell types.
Yeah.
Embryonic stem cells derived from early embryos are pluripotent, meaning they have the potential to become any type of cell in the body. They're like the ultimate blank slates, capable of giving rise to all the different tissues and organs.
So embryonic stem cells are like the master key that can unlock any door in the cellular world.
A perfect analogy. Then we have adult stem cells, which are found in various tissues throughout the body. They're more restricted in their potential, typically giving rise to cell types within their tissue of origin.
So they're more like specialized training programs, preparing recruits for specific roles within a particular department.
Exactly. For example, hematopoietic stem cells in the bone marrow can give rise to all the different types of blood cells, while mesenchymal stem cells can differentiate into bone, cartilage, and fat cells.
That makes sense. It's like having different branches of the military, each with its own specialized training programs for specific roles.
And these adult stem cells play a crucial role in tissue homeostasis, replacing cells that are lost due to injury or normal wear and tear. They're essential for maintaining the health and integrity of our tissues and organs throughout our lives.
It's fascinating to think about how these stem cells are constantly working behind the scenes, keeping our bodies running smoothly.
It truly is. And as we learn more about stem cells and how they work, we're uncovering this incredible potential for using them in regenerative medicine. Imagine being able to repair damaged tissues, or even grow new organs from a patient's own stem cells.
That's incredible. It sounds like science fiction, but it's becoming a reality. The possibilities for stem cell therapy are truly exciting, but unfortunately, as with anything in biology, there are always potential downsides. Even though cells are incredibly sophisticated systems, things can sometimes go wrong.
You're right. And one of the most striking examples of this is cancer.
Cancer, a disease that affects so many people. It's a scary word that often brings to mind images of uncontrolled growth and tumors.
You're right, cancer can be a frightening diagnosis. And it all boils down to a breakdown in cellular control, leading to uncontrolled cell growth and division.
So it's like a rogue cell that's gone off script, ignoring the rules and multiplying out of control.
Exactly. Cancer cells ignore the normal signals that regulate the cell cycle, dividing uncontrollably and invading surrounding tissues, disrupting the harmony of the cellular orchestra.
It's like a wild, untamed symphony, with instruments playing out of tune and out of time.
And this uncontrolled growth is often driven by mutations in genes that regulate the cell cycle. Those genes involved in checkpoints and growth signals. Imagine a car with a broken accelerator pedal, speeding out of control. That's what happens in cancer cells. The normal brakes on cell division are either missing or malfunctioning, allowing the cells to divide uncontrollably.
So it's like a car that's lost its ability to stop, leading to a dangerous crash.
There are two main classes of genes that are often mutated in cancer: oncogenes and tumor suppressor genes. Oncogenes are like stuck accelerators, sending constant go signals to the cell, even when they shouldn't. They're mutated versions of normal genes called proto-oncogenes that promote cell growth and division. When these genes are mutated, they can become hyperactive, driving uncontrolled cell proliferation.
So it's like having the gas pedal constantly pressed down, even when you're trying to slow down or stop.
Tumor suppressor genes, on the other hand, are like the brakes. They normally inhibit cell growth and division, acting as guardians against cancer. But when these genes are mutated, the cell loses its ability to put on the brakes, allowing for uncontrolled growth.
So it's like having faulty brakes, making it impossible to slow down or stop the car.
Exactly. And cancer often arises from the accumulation of mutations in both oncogenes and tumor suppressor genes. It's like a series of unfortunate events, each one pushing the cell further down the path of uncontrolled growth.
So it's not usually just one single mutation that causes cancer. It's more like a cascade of genetic errors that ultimately leads to uncontrolled cell division.
Precisely. And these mutations can be caused by a variety of factors, including genetic predisposition, exposure to carcinogens, like certain chemicals or radiation, and even random errors during DNA replication. It's a complex interplay of genetics and environment.
So it's a reminder that while our genes play a role, our lifestyle choices and environmental exposures can also influence our risk of developing cancer.
Absolutely. And cancer cells often exhibit other hallmarks, like the ability to evade apoptosis, that programmed cell death we talked about earlier. They can become immortal, dividing indefinitely. And they can even invade surrounding tissues and metastasize, spreading to distant sites in the body.
So they're not just growing out of control, they're also breaking free from their normal boundaries and spreading throughout the body, causing even more damage.
Exactly. Cancer is a complex and challenging disease, a testament to the power of evolution at the cellular level. But scientists are making significant progress in understanding cancer and developing new treatments, including targeted therapies that specifically attack cancer cells. immunotherapies that harness the power of the immune system to fight cancer, and gene therapies that aim to correct the genetic defects that drive cancer development.
It's encouraging to hear that scientists are making progress in the fight against cancer. It gives hope that one day we might be able to prevent or cure this devastating disease.
Absolutely. And it's a reminder of the importance of scientific research and the incredible potential for using our knowledge of cell biology to develop new treatments and improve human health.
Speaking of which, our understanding of cell biology has come a long way since those early days of peering through microscopes. From those initial glimpses of cells to the intricate molecular mechanisms we've been discussing, it's been an incredible journey of discovery.
It truly is. And it's amazing to think about all the incredible tools and techniques that scientists have developed to study cells, from powerful microscopes that can visualize individual molecules, to sophisticated genetic engineering techniques that allow us to manipulate genes and explore their functions.
It's like we've gone from exploring a new continent to mapping its intricate details and covering its hidden treasures and learning its language. And it's a journey that's far from over. There's still so much we don't know about these fascinating building blocks of life. But with each new discovery, we gain a deeper appreciation for the complexity, elegance, and resilience of these microscopic machines that make life possible.
Absolutely. And it's this sense of wonder and discovery that drives scientists to keep asking questions, keep exploring, and keep pushing the boundaries of knowledge.
It's a reminder that even though cells are tiny, they hold the keys to understanding life itself. And who knows what incredible breakthroughs await us as we continue to explore this microscopic universe.
The possibilities are truly endless.
Yeah, it really is mind-boggling to think about how much we've learned about cells, and yet there's still so much more to discover. One of the things that really struck me during this deep dive was the fact that a human gene can actually function in a yeast cell. That just blows my mind.
It is remarkable. It's a testament to that shared evolutionary history of all living organisms. At the cellular level, we're all connected. Those core mechanisms of life, from DNA replication to protein synthesis, they're so fundamental that they've been conserved across billions of years of evolution. It's like we're all reading from the same ancient genetic cookbook, with slight variations and adaptations that have led to this incredible diversity of life we see today.
Yeah, it's like we're all part of this one big interconnected family tree, sharing these ancient cellular recipes that have been passed down through generations. And speaking of interconnectedness, this deep dive has really highlighted how integrated all those cellular processes are. It's not just a bunch of isolated reactions happening randomly. It's like this beautifully orchestrated symphony of events, with each process influencing and relying on the others.
Absolutely. From DNA replication to protein synthesis to energy production, it's all interconnected. If one process goes awry, it can have ripple effects throughout the entire cell. It's like a complex machine where every gear and cog needs to be working properly for the whole thing to function.
Right. It makes you realize that cells are incredibly dynamic and adaptable. They're constantly monitoring their internal and external environments, adjusting their behavior based on the signals they receive. They're not just passively reacting, they're actively making decisions and taking action to maintain their internal balance and respond to challenges.
That's right. Cells are constantly engaged in this delicate balancing act, maintaining homeostasis, which is that state of internal stability. That's essential for life. They're like these tiny self-regulating ecosystems, constantly adjusting their activities to stay in equilibrium. And it's this ability to sense and respond to their environment that allows them to survive and thrive in this constantly changing world. It's what allows us to adapt to different temperatures, fight off infections, and heal from injuries.
Yeah, this whole conversation has really given me a new appreciation for the complexity and resilience of cells. It's amazing how these tiny microscopic units of life can carry out such sophisticated functions, and how they can work together to create the incredible diversity of organisms we see around us.
And we're only just beginning to understand the full extent of their capabilities. Every day, scientists are making new discoveries about cells, uncovering new mechanisms, and gaining deeper insights into the fundamental processes of life.
Yeah, it's exciting to think about all the unanswered questions that are still out there, waiting to be explored. What are some of the big mysteries in cell biology that scientists are still trying to solve?
Oh, there are so many. We're still trying to fully understand how cells make decisions, how they communicate with each other, how they age, and how they go wrong in diseases like cancer. We're also exploring the incredible potential of stem cells for regenerative medicine, trying to harness their power to repair damaged tissues and even grow new organs.
It sounds like there's no shortage of fascinating research questions to keep scientists busy for generations to come.
Absolutely. And it's not just about basic research either. Our understanding of cell biology has far-reaching implications for fields like medicine, biotechnology, agriculture, and even environmental science. It's a foundational science that's helping us develop new treatments for diseases, engineer more sustainable crops, and even clean up pollution.
It's incredible to think about how something so small, like a cell, can have such a huge impact on our world.
It really is. And it's a reminder that even though cells are microscopic, they hold the keys to understanding life itself. By studying cells, we're not just learning about biology, we're gaining insights into the very nature of existence.
So as we wrap up this deep dive into cell biology, I'm curious to know what stands out to you as the most remarkable aspect of this field? What really gets you excited about studying cells?
For me, it's the sheer elegance and efficiency of it all. Cells are like these tiny, self-contained universes, carrying out these incredibly complex processes with remarkable precision and efficiency. From the way they replicate their DNA to the way they manufacture proteins, it's also beautifully orchestrated. And what's even more amazing is that these processes have been honed over billions of years of evolution, resulting in systems that are both robust and adaptable.
Yeah, I totally agree. And what I find particularly fascinating is this concept of emergent properties, how these simple building blocks, atoms and molecules, can come together to create something as complex and wondrous as a living cell. It's like the whole is greater than the sum of its parts.
Right, precisely. And we're only just beginning to scratch the surface of understanding how these emergent properties arise. There's so much more to explore, so many mysteries yet to be unraveled.
Well, I don't know about you, but I'm feeling incredibly inspired to learn more.
As am I. And I encourage our listeners to do the same. Dive deeper into this fascinating world of cell biology. Explore the resources available online, take a class, read a book, and never stop asking questions.
Who knows what incredible discoveries await us in this microscopic universe?
The possibilities are truly endless.